Thank you. And I'd like to thank the event organizers for giving me the opportunity to talk today. And what I'd like to start with is that cancer incidence varies across species widely. For example, mice are prone to cancer, where elephants seem to get very little cancer. And that these differences in cancer susceptibility are due to both Sorry, is that better? The differences in cancer susceptibility are likely due to both environmental exposures and evolutionary pressures shaping an organism's life history. I also want to point out that beneficial traits such as defense against disease, such as cancer, are often costly. And that there are trade-offs between benefits early in life and disease susceptibility later in life. So this leads us to ask what factors may influence defenses against cancer across species. So first, I'd like to give some examples. This is a free-ranging male deer. What you're seeing here is tumors in their antlers. And it's found frequently enough in over 5 different free-ranging species that they actually coined the term antleroma. This is a platyfish, and this black stuff you're seeing right here is actually melanoma. Even though this disease caused reduced swimming speed and early death, it persists in natural populations. And the domestic hen. The domestic hen is actually the only non-human animal to spontaneously develop ovarian cancer. It's in high prevalence, about 30 to 35% of hens by age 3. Develop ovarian cancer. And so from these examples, I'd like to ask if we see general trends. For example, deer, the antlers are weapons that are important in competition and mating success. So this trait is important for reproductive success. Additionally, the platyfish, fish with the melanoma actually have a larger body size, and in these populations, larger males mate more often, so this trait is important for reproductive success. And then the domestic hen, they are artificially selected to ovulate and lay eggs daily. So again, this trait is important for reproductive success. So this led us to ask, could there be trade-offs between reproductive competitiveness and cancer defenses? And what I mean by reproductive competitiveness is the energy an organism allocates to reproductive effort and mating effort. And I'd like to, reproductive competitiveness is actually very important in the force of evolution of extreme morphologies and life histories, especially when the reproductive skew is high. That's in the winner-takes-all nature. And that leads to the evolution of sexually selected traits, earlier reproduction, and higher fertility. And so using a mathematical model, we explored the trade-offs between cancer defenses and reproductive competitiveness. This model was developed by Hannah Coco, and now I don't have time to go over the model. I have a handout with the whole model printed out if anyone's interested. But I briefly wanted to define some of the parameters for clarity. And we can define cancer as the end steps take the form with each occurring at a rate of k. And then we define the defense against cancer as D, where 0 is no defense to cancer. And in reproductive competition, the parameters I want to point out is alpha, which is a reproductive skew. And when alpha is greater than 1, the competition is a winners-takes-all nature. And the parameter beta is going to be the strength of the trade-off between reproductive competition and cancer defenses. There's 2 ways of dying in this model or ending our cancer-free lifespan. One is a mortality other than cancer, and the other one is cancer. That is the number of cell lineages that had passed through all the rate-limiting steps. And so with this, we wanted to vary the intensity of the trade-offs and also vary the levels of extrinsic mortality. And this allows us to explore the evolutionary viability of cancer defenses in species in different reproductive and ecological conditions. So here are the results of the model. We're looking at fitness, where yellow is high and blue is low fitness. On the y-axis is defense. Again, 0 is zero defense against cancer, and we're doing increasing reproductive competitiveness. And what we see is cancer defenses remain high throughout the increasing reproductive competitiveness until they reach a critical threshold where it dramatically declines. And if you remember, I pointed out that when alpha is greater than 1, it is in a winner-takes-all nature. So we see this, this trend to hold true for all varying levels of trade-offs and varying levels of mortality situations. However, in the high mortality situation, the cancer defenses are low to begin with. And for example, short-lived species that live in high extrinsic mortality conditions, such as mice, are likely to die of other causes other than cancer, so there's no point to invest in cancer defenses. However, long-lived species, such as elephants, probably benefit from investing in cancer defenses. We also modeled the probability of getting cancer, again varying the trade-offs and the mortality conditions. And we again see with increasing alpha, that is increasing the reproductive competitiveness, it increases the chance of getting cancer, or increases your chances of getting cancer in all the mortality conditions, again with the strongest effect in the low extrinsic mortality environment. So from these results, we started to make predictions. Males invest heavily in mating effort, and male-male competition favors the evolution of secondary sex characteristics. So we predict that these sexually dimorphic species to be subject to increased cancer risk. That includes species with extreme ornaments, large body size, and strong sperm competition. Now females, they invest in other components of reproductive effort than mating effort. We predict factors that elevate fertility may lead to an increased cancer risk. These include fertility timing, the frequency, and the litter size. Now we're proposing two potential mechanisms where reproductive competition can influence cancer risk. One is the selection for rapid proliferation. That is, if this cell has a mutation, it can rapidly increase in the population. And the other mechanism is the traditional life history trade-off between reproduction and survival. That is, energy allocated to survival comes at the cost of reproduction. To go into a little bit more detail, in rapid growth, what we find is growth factors and steroid hormones are important in the development of sexually selected traits, and they're also very important in cancer. And growth factors or low levels of IGF, which is insulin-like growth factor, are associated with lower cancer risk and may extend the time to malignant proliferation. And hormones are also associated with cancer. High levels of testosterone and estrogen have been implicated in prostate cancer and breast cancer. So we think species may actually benefit from enhanced proliferation at the cost of cancer risk. Back to the examples I provided earlier, in the deer, antler growth is controlled by both IGF, insulin-like growth factor, and testosterone. And the gene responsible for the melanin formation in this fish is a gene derived from an epidermal growth factor. It's called XMARK. So we think this male-male competition can lead to strong selection for growth and produce these elaborate characteristics or larger body sizes, but dysregulation in the system can also lead to uncontrolled growth. And our second mechanism is traditional life history trade-offs. And what I want to make a point of is that tumor suppression is a major component of somatic maintenance. That is DNA repair, immune defense, and cell cycle control. And though, how does somatic maintenance possibly influence the reproductive factors? These mechanisms are just beginning to be understood, but there is evidence in the literature for trade-offs between fertility and cancer. For example, P53 is an important tumor suppressor protein, and there's support that the family of P53s may be involved in fertility and reproduction. That includes the quality and survival of the oocytes. They're involved in early blastocyst division and important in fertilization and litter size. Another example is the BRCA gene. BRCA plays a role in DNA repair. Women with BRCA mutations have an increased risk for breast cancer. However, one study showed that these women have different reproductive success than age-matched controls. These women with BRCA mutations had a significantly more children and shorter interbirth intervals. And the KISS gene. This gene is a gene-protein-coupled receptor ligand. It's the metastasis suppressor gene that was originally named. It's important for sexual maturation, and it's also been implicated in trophoblast invasion and angiogenesis in the placenta. So if we take a step back and think, what can we say about humans, and does reproductive competitiveness influence cancer risk in humans? We think it does. We think that our model can help explain the differences in overall cancer risk between men and women. That is, men get more cancer than women. And IGF, insulin-like growth factor, is an important determinant of height in humans. Taller men are reproductively more successful than shorter men. Taller humans also have an increased risk for cancer. High testosterone levels are associated with reproductive effort and associated with prostate cancer. And also higher estrogen levels are associated with attractiveness, follicle maturation, and oocyte quality. And high cycling estrogen is associated with hormone-positive breast cancer. So in conclusion, I'd like to say that species vary in susceptibility. We think one component of it could be due to reproductive competition. Our model predicts that strong intraspecific competition can lead to high cancer incidence. And the review of the literature suggests that selecting for growth or just energy allocation trade-offs can influence both reproduction and cancer risk. And lastly, while what I laid out here could be a simplified version of probably what's going on, we need more data, especially from species with diverse life history variables, to begin to tease apart cancer susceptibility across life. With that, I'd like to thank my collaborators and co-authors of my paper, and like to say that this project was what happens when a cancer evolution group and an extreme traits group get together in Berlin and drink a lot of coffee. So, thank you.
Thanks. I think we have time for a question. Or wherever you want to take the mic.
In the winner-take-all situation, there are many species in which the males are very competitive, in extreme cases, like in hippos, fighting often to the death, which is not often the case with other species. Are there differences in relative cancer rates in males versus females? For example, do you expect a higher than normal level of cancer rates in male elephants compared to other male animals, and relatively low cancer rates in female elephants compared to female other animals?
Yeah, that's a good question. Yeah, we would predict that you would see more cancer in a male elephant that's extremely competitive. However, it's very hard to find data for this in the natural populations, especially in extreme competitive competitions, because these males are dying, they're mating to death. So they are likely to die in competition rather than of cancer.
Okay, thanks very much for that. We need to move on.